Hi everyone. So today we'll take the topic CRISPR technology, CRISPR's biotechnology. So why it is in the news? India approved a 5-year project to develop CRISPR to cure sickle cell anemia. We'll see in detail. So for now you have to understand India approved a 5-year project under the CRISPR biotechnology. So see, this is a probable mains question under science and technology. The GS paper 3, so we have the topic science and technology. This is a very important topic for this mains because in future many more developments are going to come on this technology and many more advantages because of this technology. It is going to change the future of the genetic engineering, that too in a very faster way. So how we are going to take this topic? What is CRISPR technology? Who developed it? How this technology works? What are the advantages and benefits of this technology, and what are the ethical dilemmas that were involved in this process? So before taking this, I'll explain you what is going on, how this works, and then we'll go back to the sentences so that you'll have a notes here itself. Now, CRISPR, what is here CRISPR? Clustered, regular, interspaced, short, palindromic repeats. So what is here? See, clustered, what we are saying, regular, interspaced, there is a space over here, palindromic. Palindromic means it will be repeating, ACTC, ACTC. So even from this side will be the same, even from this side it will be the same. So palindromic repeats. So this is the definition of this CRISPR. So before going to the technology, I want you all to understand what is DNA and what is RNA. I'm not going to take in detail about this, but what is needed for the topic, that only we discuss. What is DNA? This DNA will carry the genetic information, right? So it's like a disk where we'll have all the genetic information. What this RNA will do? This RNA will produce proteins. This RNA will produce proteins. What this protein contains? The protein contains the instructions, which were the instructions given by this DNA or in the genetic material. What are the instructions that we have? That will be in this proteins. The protein carry these instructions. So if you take this as a floppy or a disk, this RNA will be a reader. So it will read this genetic material. Based on that, it will produce the proteins. Okay. So now, so this CRISPR technology is a very old one. We know this long back, but the technology to take the out of it and develop an easier way for the genetic engineering, that was the recent one. So first we'll see, first we'll understand what is CRISPR technology in bacteria. Initially they found it in bacteria. We know that earlier also, the CRISPR technology. But taking the advantage of it, that is a new one. So what happens in bacteria? See, this is our DNA. The small bacteria, single cell bacteria, Anemia or E. coli we can take. So this is a DNA. Okay. So now what happened? A virus came. So now this bacteria was attacked by a outside virus. It entered into the bacteria's body. Now what happened? The Cas protein which was existing in our body, what it will does, it will go, see here is a virus, so a small virus was there. This Cas protein will go over here and take the DNA, the DNA material of this virus, and it will cut this line and keep the DNA material of it over here. See, I'll explain again. See, this is a DNA material of this white ones, the DNA material of the bacteria. This Cas protein will take the material from the virus that will affect the bacteria. It will take the material from the virus and it will insert that into the DNA material of the bacteria. So what does it mean? Now this bacteria has a component of the virus which was done by this Cas protein. Now what happens? This one will produce more RNA. What is RNA? What is RNA, guys? So this RNA is protein which carries the instructions or whatever that is replicated here. It will be whatever the genes carry, this gene carries in instructions that were there in these proteins. Now what will happen? This Cas9 will take this protein. Now again a virus came into the bacteria's body. Now virus came again into the bacteria's body. Now this Cas9 is waiting. So now what it will do? It will attack the virus and it will destroy. So this is a basic immune system that existed in a bacteria. See, I'll explain you again. Please listen. This is a DNA of a bacteria. It has genes. Now, when a virus attacked the bacteria, so this Cas protein will take the gene material of the virus, and it will cut this DNA chain and insert this into here. This is from the virus body. This is from bacteria and this is from the virus body. Now, what will happen? This Cas protein brought this and put it over here. So now they will produce RNA. So what is this RNA? It contains the instructions from the gene, this DNA, whatever the instructions on this DNA, it will be in the RNA. So what is the instruction? It had the gene code of the virus. So this will be taken by the Cas9. So now when it attacks, when the virus comes again, so this will attack the virus and destroy this. This is the immune system. So initially when the virus attacked the bacteria, so they were able to take the DNA from the virus and inserted it into the Cas9 protein. The Cas9 protein, they inserted it, and then later on again when the virus attacked it, it already had it in its DNA. So when this came over, so it will destroy it. That is how the technology works in a bacteria's body. This is an immune system. How we brought that into the CRISPR technology? That is how we are going to take the part. Now listen carefully. So now here we have a DNA. So DNA will have two strings. Two strands. Now what we have to do? We have to take a Cas9 protein. Do you remember this Cas9 protein? This yellow one. So what we have to do? In the Cas9 protein, we will have a DNA. The Cas9 protein will have an RNA protein. What that protein contains? That protein contains information related to the DNA. So that is matching DNA. So here, I want to attack this DNA. So what I have to do? In the Cas9 protein, I will make a RNA which is called guide RNA that is matching with this DNA, which is called guide RNA. So when these two are matching, so what will happen? So it will come, see, the things are very clear over here. This RNA contains the material that is matching with this DNA. So when they are matching together, this guided RNA will come over here. And what is the purpose of this Cas9 enzyme? What is this Cas9 enzyme did? It acts as a scissor. So it will cut the DNA over here. So see here, if you observe, so this is how the DNA fragments were cut. So now what will happen? Now I want to change the gene. When it was cut, our body will try to repair it. When it was repairing, so instead of allowing the old one, we will substitute it with the new gene. Guys, I'll explain you again. So suppose a gene is not, the people are having, because of the genes they are having sickle cell anemia. What is sickle cell anemia? I'll tell you this example so you'll understand very clearly. Sickle cell anemia, let's think it's in our body. So what is it? Red blood cells in a body will live for 120 days. Red blood cells will live in a body for 120 days. After that, they will regenerate. Okay, they will be replaced. But if for some people, it will last only for 12 to 10 days. For some people, it will last for only 12 to 10 days. So then what happened? They need to be regenerated early. It causes an anemic problem. So that is sickle cell anemia issue. So let's think this whatever the strand over here, it is a sickle cell gene. Now what we are trying to do? So I have to make an RNA. What is the RNA? RNA will contain the proteins. We have to make an RNA. That RNA should match with this DNA. We can make that within a system. You can make that. So we'll make an RNA, artificially we'll make an RNA that will match with this DNA we made. Now we'll keep this RNA in a Cas9 protein. In that protein, we'll keep this RNA. So this is, which is called guide RNA. What does this guide RNA does? It is matching with this DNA. So it will go and connect over here, this guide RNA. So then what will happen? So they have to, this Cas9 protein will cut this DNA. When it cuts, they acts like a scissors. So when it was cut, it will have the fragments, DNA fragments cut by Cas9. And then what will happen? So we will use so while repairing, we'll use an inserted DNA for the edited section. So I'll give you another picture over here so you can understand it. So this is a DNA strand. So in the DNA strand, this is the targeted one. So to match this targeted one, I have to produce a Crispr molecule. What is this? This is a Crispr molecule. So what does it contain in the Crispr molecule? So the guide RNA. What is the purpose of this guide RNA? The guide RNA takes this Cas9 protein to attach this, because this has an idea which DNA it has to attack. So then it will go and attach. So this Cas9, what is the purpose of Cas9? It will cut the molecule. So then what will happen? Then a new one can be attached. Only purpose of this guide RNA is to make it attached to the target DNA. When this molecule reach the target DNA, how it will reach? Because we matched it. Then it will cut and will have a strand. Okay, so here we can understand. We will go through it once. As I told you earlier, clustered regularly interspaced short palindromic repeats. So it is simple and far more accurate when compared to the earlier genetic engineering. So now it is like a cut and copy paste. So by using Cas9, we will cut it. So what we'll do, we are copying an DNA into a guided RNA. We will go and attach, it will copy that, and we will paste a new one. So that is how this works. So who developed it? So these people developed it and they achieved a Nobel Prize in Chemistry in 2020. So we have saw how this works, and we have seen. So the RNA molecule is programmed to locate this sequence to the targeted DNA. Whatever the DNA we want to target, that was there in the RNA molecule. And what is the purpose of this Cas9? They'll act as a genetic scissors. They will break the DNA strand. So when this RNA guides the Cas9, what is the purpose of guide RNA? It will go and attach the targeted DNA. Cas9 will cut it and will supply it with the correct sequence. So we can change it. So that is the same case with the sickle cell anemia. So what are the advantages? So the major advantages of this instrument or with this technology is diseases and disorders that were genetic in nature can be cured. So it could be blood diseases, majorly sickle cell, eye diseases, color blindness, HIV, diabetes, liver and heart diseases, many more applications for this genetic engineering. So we can see here, so it is targeting in medicines we can develop for the tumors, especially the cancers which were genetical. So we also can have in biofuels, we also can have in even for the crops, industrial products, we have huge opportunities for this technology. Whereas challenges, we'll see them. So first we'll see the recent developments. So many clinical trials were undergoing, especially in the US. So much more development took place under this technology. So under diabetes and blood disorders and inherited eye diseases. So in India, that was in the news, in India, we allowed this technology for the sickle cell anemia. So what are the ethical dilemmas? Issue of designer babies. So what is the here issue of designer babies? We have seen it is in China. So how a gene in children were changed so that they won't be attacked by HIV. So this could be an probable issue. So people can give different characters. If they change the gene, so they can, then it will be an artificial process, not the natural selection. So definitely it is not a foolproof technology, even though it is an easier one, fastest one and most accurate one, but it is not a foolproof technology. And the people want it to be a therapeutic intervention rather than a preventive one. So they want to be a therapeutic. The changes in the genetic sequences will remain in the individual and not passed on to the offsprings. It is therapeutic. So what could be the possible question for this? So it's pretty clear. Directly they'll ask us what is the technology, what are the advantages and what are the ethical dilemmas that were involved in this biotechnology. Okay. Thank you guys. Please attempt this question. Thank you.